Hello, my name is Professor Hudson, and you are about to view narrated PowerPoints for Chapter 12 and Chapter 13. We will finish talking about Chapter 12 with what are mutations and how do they occur. In Chapter 13, we will talk about gene regulation. These two chapters are for Module 6, Part 2, DNA Biology, Mutations and Gene Regulations. Let us begin. So let's start talking about what are mutations and how do they occur. Mutations are changes in the base sequence of DNA caused by mistakes during replication or by various environmental factors. The effects of mutations depend on how they alter the codons of messenger RNA. Mutations can affect protein function in many ways. Some different categories of mutations include inversions, where chunks of DNA are flipped around, translocations, where chunks of DNA are moved from one chromosome to another, deletions, chunks of nucleotides are removed, and duplication, chunk of DNA segments can be repeated multiple times. This figure is not in your textbook, but it does show some of the categories of mutations that were mentioned previously visually. As mentioned already, inversions and translocations occur when pieces of DNA are broken apart and reattached within a single chromosome or to a different chromosome. Notice in this figure, in the inversion, the gene sequence was supposed to read A, B, C, D, E, F, G, with each letter corresponding to a gene. However, two of the genes flipped around such that now the genes read A, B, D, C, E, F, G. With translocation, if you noticed, genes F and G have moved from one chromosome, the pink one, over to the green chromosome. That is translocation. Inversions and translocations may be benign if entire genes are merely moved from one place to another. However, if a gene is split in two, it will no longer code for a complete functional protein. There is a severe type of hemophilia that is caused by an inversion in a gene that encodes a protein required for blood clotting. Also in this figure, they show you an example of deletion. A deletion mutation occurs when one or more nucleotides are removed from the gene sequence. In this case, the chromosome in pink has lost the A gene. And duplication occurs when a segment of DNA is repeated multiple times. In this illustration, if you notice, genes D and E occur twice. This figure is illustrating inversion by itself. As mentioned previously, inversion is where a gene or several genes are flipped around. In this example, in the pink chromosome listed here, it was supposed to read C, D, E, but for some reason, those three genes flipped around. Thus, a segment of this chromosome is inverted. When crossing over occurs between the one from mom, which is this pink one here, with the one from dad, which is the blue one, both chromosomes can end up having a duplication of some segments and a deletion of other segments. This figure is highlighting translocation, and it also highlights two syndromes that can occur when translocation occurs. Remember, translocation is where a segment of DNA is moved from one chromosome to another. In Alagille syndrome, a segment of one chromosome is moved to another one. In particular, the chromosomes involved are chromosomes 2 and 20. They exchange segments of DNA. This causes Alagille syndrome. This syndrome has distinctive facial features, and oftentimes people do not realize that they have the syndrome until they have a child with the syndrome. Down syndrome, in a small percent of cases, also involves a translocation between chromosomes. In this case, a piece of chromosome 14 moves to chromosome 21. These people have Down syndrome, but they have a much milder form because they do not have a whole extra copy of chromosome 21. They just have a little piece of chromosome 14 stuck on one of their 221 chromosomes. Deletion. This slide is illustrating a couple of syndromes that can occur when DNA is lost. And it depends on what genes are lost, what type of syndrome you can end up with. In this example, Williams syndrome is caused by a deletion in chromosome 7. And then this figure is showing 4 children who are not related who have Williams syndrome. They have the same appearance, health, and behavioral problems. Children with this syndrome look like little elves or pixies because they have turned-up noses, wide mouths, a small chin, and large ears. Although their academic skills are poor, they exhibit excellent verbal and musical abilities. Another syndrome that is caused by deletion is pronounced cri du chat, which also stands for cry like a cat in French. This is caused by a deletion of chromosome 5. Babies who have this syndrome often have a cry that sounds like a kitten meowing, hence the name cry like a cat. Duplication is where gene sequences get repeated. There's a syndrome called MDUP15 where people with this syndrome have poor muscle tone, mental retardation, and other related health issues. This is caused by a duplication of genes and an inversion. People suffering from MDUP15 syndrome will have poor muscle tone and often will be autistic. Note, these examples and syndromes are not mentioned in your book. However, still please be familiarized with them. The examples in this lecture are used mainly just to give you a snapshot of some of the things that can happen when chunks of DNA are duplicated, inverted, or undergo translocation. Now let's talk a little bit more about mutations and some of the types of mutations that more commonly occur in DNA. A mutation is any change in the nucleotide sequence of DNA. Mutations can change the amino acids in a protein. Mutations within a gene can occur as a result of point mutations. Oftentimes, these point mutations fall into one of 3 categories: base substitution, nucleotide deletion, or nucleotide insertion. Also, it's worth mentioning that nucleotide deletion and nucleotide insertion are often referred to as frame shift mutations because the deletion and insertion can throw off the reading frame for synthesizing the protein. In base substitution, one base is replaced with another. In nucleotide deletion, a nucleotide is lost. In nucleotide insertion, a nucleotide that wasn't there is added. This slide illustrates the 3 types of mutations talked about on the previous slide and their effects. The first figure, A, talks about base substitutions. When a nucleotide is substituted, we call that a point mutation. And this occurs when an incorrect nucleotide takes the place of what was supposed to be there. A point mutation in the form of a substitution sometimes does not change the amino acid sequence of the protein. Because many amino acids are encoded by more than one codon, the mutation may cause the same amino acid to be added. An example of this happening is there is a known point mutation in the beta globin gene for hemoglobin, which causes CTC to change to CTT. However, when the final protein is made, the amino acid glutamate acid remains unchanged. So there's a point mutation in the form of a substitution in the DNA. However, the protein is unaffected. Also, you can have deletions and insertions. Remember, a deletion mutation occurs when one or more nucleotides are removed from the gene sequence. In some of the previous slides, we talked about when whole chunks of DNA were deleted or inserted. Here we're referring to typically just 1 to 3 nucleotides deleted or inserted. When a nucleotide is deleted, the radium frame can be thrown off, and this typically will lead to problems with the protein. Similarly, with an insertion, you have a nucleotide added, maybe 1 to 3 nucleotides added that weren't supposed to be there. This also will throw off the reading frame. Deletions and insertions are illustrated in part B and C of this figure. When talking about point mutations, a common everyday example of a disease that's caused by point mutations is sickle cell anemia disease. If you look at this figure, it illustrates that in normal hemoglobin, the DNA should read CTT. The hemoglobin gene in DNA encodes for the hemoglobin protein, which carries oxygen around in the bloodstream. CTT in the DNA gives rise to messenger RNA with the sequence GAA, which encodes for glutamic acid. However, people who inherit the mutation for sickle cell anemia, their DNA will read CAT instead of CTT. The CAT gives rise to messenger RNA that reads GUA, which then gives rise to a protein that has a valine amino acid where they should have had a glutamic acid. This causes the protein hemoglobin to misfold, and it does not carry oxygen as effectively. This leads to the symptoms of sickle cell anemia disease. This is an example of a point mutation at the level of the DNA where a single nucleotide is changed that ends up changing an amino acid in a protein. It's a point mutation at the level of the DNA, and we call this a missense mutation at the protein level, in that we received an amino acid that would not have been there had we had the correct DNA. This figure is also illustrating what was discussed on the previous slide with sickle cell anemia disease in humans. And it's showing what normal red blood cells that have the normal hemoglobin protein with the glutamic acid amino acid look like, and then what sickle cell red blood cells look like that inherited the mutated incorrect valine allele. In the next segment of the narrated PowerPoint lectures for this module, we will continue talking about the effects of mutations on protein activity.